In this tutorial, we're going to look at regulation of gene expression, and we're going to define what a lac operon is, and also the operation or how a lac operon is able to operate in order to produce the proteins. So this is what we're going to look at. But before we can go any further, we're going to understand what regulation of gene expression is, and also what gene expression is all about. So let's get started. So we're going to start with gene regulation. Before we can define what gene regulation is all about, let's try to understand what the word regulation means. So to regulate is just to control. When we say a certain thing is regulating another thing, it simply means that that thing is controlling another substance or that other thing. So that is what regulation just means. So now let's try to understand what gene expression is. So when you're talking about gene expression, gene expression is just the process by which the information in a certain gene is able to lead into the production of proteins. So that is what gene expression is. So we are saying process by which information in a gene leads to the production of proteins. During the central dogma of molecular biology, we defined the stages which are essential to be carried out in order to lead to the production of proteins. We said that first of all, the information in a DNA has to be made in several copies. This DNA has to make another copy of DNA. By the process, we said it is DNA replication. Replicate is just to make a copy of another DNA. Then this information in a DNA has to be converted into RNA, and specifically we said it is the messenger RNA. So DNA makes a copy. It is not the entire DNA, but it's just a segment or a small portion of DNA that is able to be transcribed into mRNA or the messenger RNA. And the process, we said, it is transcription. Then this information now, which is now contained in mRNA or the messenger mRNA, has to be translated, has to be made into proteins. Okay? And the process we said, that is now translation. So we need to understand that at each and every portion or at each and every stage of replication, transcription, and also translation, there has to be the regulation. So this regulation can be done or carried out during replication. It can be carried out during transcription, or rather during translation. So that is very important for us to appreciate. Now let's look at the regulation of gene. What does it mean when we talk about regulation of gene? So regulation of gene. So when you're talking about the regulation of gene, you're just referring to the processes which are involved to increase or decrease the production of proteins. You're just trying to talk about process or processes rather involved to either increase or decrease the production of proteins. You see, there are times when the body does not need so much proteins, and if the body tends to produce more than what is required or too much proteins, those proteins can just be wasted. They can't perform any function. So there has to be this regulation to control, to regulate the amount of proteins which has to be produced at a certain time, and when and where should they be produced. So that is the importance of this regulation of gene. Now let's try to understand now the broad theme. What is now regulation of gene expression? So we combine now expression, gene expression. We've said it's just a process by which the information in a gene leads to the production of proteins, and we've defined what regulation of gene is. We've said it is just a process that has to be carried out either to increase or decrease the production of proteins. Now, what is regulation of gene expression? Regulation of gene expression. So now when you're talking about regulation of gene expression, you're just referring to the several steps that has to be involved either to increase or decrease the production of a specific protein. So you're just referring to the steps now. Steps that has to be involved to increase or decrease the production of proteins. So we need to appreciate that concept. It is very important. But before you can talk about the operation of a lag operon, let's first try to understand the classes of genes. So basically there are two classes of genes. There are two classes of genes. The first one we have is what are known as the regulatory genes. So we have the regulatory genes. Secondly, we have what are known as structural genes. So these are the two major classes of genes, regulatory genes and also the structural genes. So we need to understand that the regulatory genes, these are the genes which are involved in regulation. They are genes which are involved in controlling. Okay? Let's try to talk about each class of gene. Let's start with regulatory genes. So we are saying that these are the genes which are involved in regulation of the production of proteins. Okay, so these are genes which control the production of proteins. And these regulatory genes, there are too many. About 98% of the genes we have, they are regulatory genes, and only 2% are structural genes. So let's try to talk about structural genes. So what are structural genes? Structural genes are these genes which are able now to code for the proteins. Okay, so I'm saying these are actual genes. They are actual genes that code for proteins or that form proteins. Okay, so it's just one and the same thing, to code or to form proteins. And we've said since 98 rather percent of the genes we have, they are regulatory genes, it implies that only 2% of those genes are structural genes. So how am I going to find 2%? You just say a percent is out of 100, right? So we have 100%. Now you subtract the 98%, which are regulatory genes. You remain with the 2%, which are now structural genes. So it is very much important for us to appreciate that concept. Now let's look at the importance. What is the importance of gene regulation? The importance of gene regulation. So gene regulation has several importances. One of the importance is that instead of producing too much genes, which can lead to the production or the usage rather of too much energy, when you are able to regulate, it saves energy. Okay, when the body is able to control that at this particular time we don't have to produce too much proteins, so the proteins production has to decrease. In that way you'll be able to save energy. The body is able to save energy by regulation. Okay, the other thing is, when there is gene regulation, it ensures that the proteins, they are made when they are required and where they are required. Okay, so ensures proteins are made when, okay, let me get rid of this, when or where required. So it is very important for gene regulation to take place. Now let's try to understand the classes of these genes. So let's have a gene. Okay, so when you're talking about a gene is just a portion of DNA that is able to control a specific characteristic. Okay, we are saying it is just a portion of DNA that controls a specific characteristic. That is what we call a gene. Okay, so we've said there are two types of genes. So let's have this portion here, then we have another portion this side. Okay, so this portion we have here, this is the portion which we call, or which we are calling rather, regulatory gene. Okay? Then this other portion here, this is the what? The structural gene. So this regulatory gene is the one which is able to carry out the regulation or the control of the production of proteins. Now this structural gene is the actual gene which codes for the proteins. Okay, so we need to appreciate that that concept. It is very important. Now when you're looking at this structural gene, okay, so few bases away from the structural gene, or just here, few bases away at this portion here, we have the coding gene. Okay, we have the coding genes here at this portion. So we need also to understand that few bases behind this coding gene, we have what is known as the regulatory sequence. Okay, so we have the regulatory sequence, just few bases behind the coding protein, okay, or the protein coding gene. So one thing I want us to understand is that when you combine the regulatory gene and also the regulatory sequence, it forms what is known as the regulatory DNA. Okay, this is how the regulatory DNA is formed. So what are we saying? We are saying that if you combine the regulatory gene and also the regulatory sequence, this is able to produce what we are calling the regulatory DNA. Okay, so and we've said that this regulatory sequence is just few bases behind the coding gene or the protein coding gene. So we need to appreciate that concept. It's very much important. Now let's look at this RNA polymerase. So if you have the RNA polymerase, so let's have a strand like that. So this side, we said we have the regulatory gene, then we have the structural gene. One thing I want us to understand is that the RNA polymerase binds to the regulatory sequence. So we have the regulatory sequence here. So this is where the RNA polymerase is going to bind. So it just binds to the regulatory sequence. The RNA polymerase will understand it better, how the binding occurs. Specifically, this RNA polymerase binds to what is called the promoter. Okay, but we'll understand it in this same video at a later stage. For now, let's appreciate the concept that RNA polymerase binds to the regulatory sequence. And after it binds, let's say you have a certain portion there, A, and another portion there, B. Let's say A and B, they are genes. Okay? Maybe let me use a different ink. So let's say we have A there, we have B there. So these are genes. When RNA polymerase binds to what is known as the regulatory sequence, it is now able to transcribe. It moves, and after it moves, it is able to transcribe the information which is coded in this gene A and also the information coded in the gene B. Okay? Now, when it transcribes these, it is able to convert these into RNA. And we understand that this RNA is what leads to the production of proteins. Proteins come from RNA. So that is very much important for us to appreciate. Okay? And one other thing I want us to understand is that there is what is known as the main regulatory gene. Okay? Just behind the protein coding gene, I can say 1,000 bases away. 1,000 bases away from the protein coding gene. So the protein coding gene was here. Protein coding gene. Just 1,000 bases away, we have what is known as the main regulatory gene. So the main regulatory gene is 1,000 bases away from the what? The coding gene. So it is very much important for us to appreciate that concept. Now, what is the importance of the regulatory gene? We've said that the regulatory gene is the one which is able to synthesize what are known as the regulatory proteins. Okay? This one synthesizes the regulatory protein. So it synthesizes the regulatory proteins. That is the importance of the regulatory gene. Now, what is now the importance of this regulatory protein? So this regulatory protein is able now to tell the RNA polymerase to bind to the regulatory sequence. Okay? The regulatory protein, which is produced from the regulatory gene, it is able to tell the RNA polymerase to bind to the regulatory sequence. Okay? So it tells the RNA polymerase to bind to the regulatory sequence. So that is the importance of the regulatory protein. So the regulatory gene produces the regulatory protein. The regulatory protein is the one which is able to tell this RNA polymerase in order to bind to the regulatory sequence. Because when RNA polymerase does not bind to the regulatory sequence, there won't be any production of proteins. Okay? It is until it binds to the regulatory sequence, then it moves and transcribes whatever genes has to be transcribed. Now, the process goes like this. When this RNA binds, when this RNA polymerase during the discussion of the central dogma of molecular biology, we said that the RNA polymerase is an enzyme that is able to help in what is known as the process of the transcription of DNA into RNA. So we said it is an enzyme. So this RNA binds to what is known as the regulatory sequence, and it is able now to increase or maybe decrease the formation of the conversion of mRNA. Okay? So this RNA polymerase, after binding, will either increase or decrease, will increase or either decrease the forming, or should I say the conversion, okay? Maybe the conversion of mRNA, which is the messenger RNA. So this is what we need to understand. So in the next video, part 2, let's explore now the operation of the lag operon.